Hello everyone. Today we will talk about proteins. So the term protein was given by Berzelius in 1538. So Berzelius, he first of all used this term. Actually it's a Greek word, proteos, which means first. So what are proteins? These are the most complex macromolecules which are really essential for the proper structure and metabolism of the biological cell. Remember, all proteins are polymers of alpha amino acids. Alpha amino acids. Now, what are amino acids? Amino means amino group, NH2 group. Acid means carboxyl group. So, what are amino acids? Simply, these are the molecules which contain one amino group and one carboxyl group. So depending upon the number of amino groups or the number of the acid group, carboxyl group, we can classify the amino acids as neutral amino acids, acidic amino acids, as well as basic amino acids. So it is just on the basis of the number of the functional groups, whether amino groups are greater than or more than the carboxyl group, or carboxyl groups are more than the amino groups, or are they in equal number. So let us start with the acidic amino acids. So simply the amino acids in which carboxyl groups, the number of carboxyl functional groups are more than the basic group, that is amino group. So they are called the acidic amino acids. For example, glutamic acid, aspartic acid. Number 2 is the basic amino acids. So amino acids in which the basic part, that is amino groups, number of amino groups are more than the number of carboxyl groups. So they are called the basic amino acids. For example, lysine. Lysine is a basic amino acid. Number 3 is the neutral amino acid. Means the amino acids in which the number of carboxyl groups are equal to the number of the amino groups. So for example, alanine, lysine, alanine and glycine. These are the neutral amino acids. Now again we can further classify amino acids as essential as well as non-essential amino acids. So what are essential? Now there are total of 20 amino acids. Out of these 20, there are few amino acids which can be synthesized by our body. These are called non-essential amino acids. And there are 10 amino acids which our body cannot synthesize, so we need to take them regularly in our diet. Such amino acids are called essential amino acids. So essentials, we need to take them in our diet, and these are 10 in number. Non-essential, our body can synthesize them, so whether we take or we don't take them in our diet, it doesn't matter, because body can actually synthesize these amino acids. So that's all about the essential and non-essential amino acids. Both of them are 10 in number. 10 essential and 10 non-essential amino acids. So here is the complete classification of amino acids, essential as well as non-essential amino acids. Now next is, what are the physical properties of amino acids? Now amino acids are usually colorless, crystalline solids. They have high melting point. Generally they are water soluble because of intermolecular hydrogen bonding with water. The most importantly, they have Zwitter ion structure. Zwitter ion structure. So what is this Zwitter ion structure? Zwitter ion structure means that the proton can be transferred from the carboxyl end to the amino site. As a result, NH2 will become NH3 positive, whereas the proton, that is COOH, which has lost the proton, gets converted into COO negative. So as a result, on one amino acid, there is one side positive and one side negative. Such a dipolar structure is called Zwitter ion structure. So alpha amino acids have dipolar ion structure or Zwitter ion structure due to the transfer of proton from carboxyl terminal from C terminal to the amino end, that is N terminal, within the same molecule. Within the same molecule. After this, let us now come to the chemical composition of proteins. Now see, we have started. What are proteins? Proteins are nothing. These are the most complex macromolecules essential for the complete structure and metabolism of the biological cell. Now, chemically, proteins are made up of alpha amino acids. Actually, proteins, they contain carbon, hydrogen, oxygen and nitrogen. Now, besides these, there can be sulfur, phosphorus, iodine, zinc, manganese, iron, etc. Now, when we hydrolyze these elements or when we hydrolyze these proteinaceous molecules or proteins, they decompose first of all to give a peptide. Then on further hydrolysis, they give alpha amino acids. So what is a peptide? How are peptides formed? Peptide, it is a linkage between the two amino acids. Linkage between two amino acids. So peptide linkage is CONH linkage. Look at the second diagram in this slide. CONH linkage is called the imide or simply the peptide linkage. Amide or peptide linkage. So the two amino acids can be connected by the loss of water molecule. The carboxyl terminal will be losing OH and the amino terminal will be removing the hydrogen in the form of water. As a result, the two amino acids, they connect with each other via amide linkage or the peptide linkage. So a dipeptide is formed by the union of two amino acids, whereas a tripeptide will be formed by the union of three amino acids. For example, here in this tripeptide, we have the first glycine, another glycine molecule, and the third is also the glycine molecule. Now these three glycine molecules have joined together via the peptide linkage, and such a peptide is called a tripeptide. Tri means three. So three amino acids have joined with each other. Now see, we can classify these also like oligopeptides, polypeptides, and then come the proteins. So depending upon the number of amino acids present, the peptides have been classified as oligopeptides, polypeptides and proteins. Oligopeptides means they contain 2 to 9 amino acids, whereas polypeptide means they contain 10 to 100 amino acids. But what is a protein? A protein contains more than 100 amino acids. So polypeptides are simply the peptides containing up to 100 amino acids, whereas the proteins contain more than 100 amino acids. 100 amino acids. Next is, what is isoelectric point of alpha amino acid? I told that amino acids have got Zwitter ion structure. Zwitter ion structure means they are dipolar. Dipolar means one end is having negative charge, other end is having positive charge due to the migration of hydrogen from carboxyl to the amino group. That means when a solution containing charged ion is subjected to the influence of electric field, then definitely the negative ions will migrate towards the positive electrode, whereas the positive ions, they migrate towards the negative electrode. Now, if in the acidic medium we put alpha amino acid, they exist as cations, so they will migrate towards cathode. Now, if we put them in the alkaline medium, alpha amino acids, they will be behaving like anion and they will migrate towards positive electrode or anode. But at a certain pH value, the amino acid, they do not migrate towards any of the electrodes and they will exist as a neutral dipolar ion. So the pH at which a particular amino acid does not migrate towards any of the electrode under the influence of the applied field, at that point, that pH is called isoelectric point. Different amino acids have got different isoelectric point. For example, alanine. Alanine has got isoelectric point 6.02, whereas arginine, it has 10.70. One more thing to remember about the amino acids, that is, amino acids may have D form, capital D form or L form, depending upon the configuration about the chiral carbon center. So, if the now in the Fisher projection, we show carboxyl group at the top position and the CH3 group at the bottom position. Now, it all depends on NH2. If NH2 group will be towards the right side, we will call it as capital D, means D alanine, let's say. Now, if let's say ki NH2 group is on the left hand side, we will call it as the L form, capital L form. Since amino is on the left hand side, so it will be L form of alanine. But if NH2 group is on the right hand side, it will be the D form of alanine. Let us now come to the classification of proteins and the structure of proteins. So let's start with the structure of proteins. I told that proteins are quite complex. So their structure can be discussed under 4 headings. Number 1, primary structure. Number 2, secondary structure. Number 3, tertiary structure. And number 4 is the quaternary structure. So primary structure of protein. It simply tells us the sequence in which the amino acids are linked to one another. So that means primary structure is only the sequence of amino acids. If there is a change in the sequence, it may lead to a different protein synthesis. So it all depends on the sequence. Number 2 is the secondary structure of protein. Secondary structure. Now, secondary structure of protein, it refers to the long flexible peptide chains of protein that are folded into the relatively rigid regular conformations. That means it simply refers to the conformations which the polypeptide chains assume as a result of hydrogen bonding between CO and NH group of different peptide bonds. Now, depending upon this linkage, there are 2 possible structures for the secondary structure of proteins. It all depends on the size of the alkyl group. So, depending upon the size of R group, alkyl group, the protein will acquire alpha helix or the beta pleated structure. Let us talk about alpha helix structure. So if the size of alkyl group is quite large, then the hydrogen bonding occurs between CO group of 1 amino acid unit and NH group of 4th amino acid unit within the same chain. As such, the polypeptide chain coils up into a spiral structure called right-handed alpha helix structure. So, this type of structure is present in our hair, in our muscles, and of course in the wool. These proteins are quite elastic. See, our hair, they do have elasticity. And when we stretch alpha helix structure, then weak hydrogen bonds causing alpha helix, they will break. So that's why the length of our hair, it gets increased. And once the tension is released, hydrogen bonds are again formed and it gives back the original helical shape. Let us talk about now beta pleated structure. Beta pleated structure. So what is this beta pleated structure? Beta pleated structure is also called flat sheet. Now if size of R group, alkyl group is relatively small, then the polypeptide chain, they will lie side by side in a zigzag manner with alternate R groups on the same site situated at fixed distances apart. Now 2 such neighboring chains will held by intermolecular hydrogen bonds. And when number of such chains are interbonded, this leads to formation of a flat sheet-like structure. These chains, they may contract or bend, but it all depends on the size of R group. As a result, the sheet bends into parallel folds to form pleated structure known as beta pleated sheet structure. Now, for example, silk. And such proteins are not elastic. Silk is a non-elastic protein. Silk. Coming to the tertiary structure. The tertiary structure of a protein refers to its complete three-dimensional structure, which arises due to folding and superimposition of various secondary structural elements to give the molecule a spherical shape. Spherical shape. Now, on the basis of the tertiary structure, we can further classify the tertiary structure as fibrous protein and the globular protein. But before that, the important term here is what type of forces stabilizes the secondary and the tertiary structures. So these are hydrogen bonds, disulfide linkage, van der Waals forces, and electrostatic forces of attraction. Now let us talk about the difference between fibrous and the globular protein. First of all, fibrous protein. Now when the polypeptide chains, they run parallel or anti-parallel and they are held together by hydrogen and disulfide bonds, we call it as a fibrous structure. Whereas when chains of polypeptide, they coil around to give a spherical shape, then we call it as a globular protein. Fibrous proteins are insoluble in water. For example, our hair, they are insoluble in water. But the globular proteins are usually soluble in water. For example, albumin, hemoglobin, they are soluble in water. Coming to now is the quaternary structure. Quaternary structure of protein can be composed of two or more polypeptide chains called subunits. Now the spatial arrangement of these subunits with respect to each other is called the quaternary structure of the protein. Quaternary structure. Now after this, let us talk about the shape and the function. Now shape and function will decide the function of the protein. Means how that protein will act, whether that protein is involved in the structural material or as a transporting agent or in the metabolic regulation. It all depends on the structure of the protein. So structure of protein is based on the shape and the sequence of the amino acids. Next is, what is denaturation of protein? Now a protein has got a specific biological activity. And when a protein has that activity, it is called a native protein. But when the native protein is subjected to some physical changes like change in the temperature, change in the pH or some other factors, then it leads to denaturation. Denaturation is due to breaking of hydrogen bond, as a result of which globules unfold and helix get uncoiled, and protein loses its biological activity. Now next is, during the denaturation, only the primary structure is intact. The secondary and the tertiary structures are lost. Next is renaturation of protein. Now some proteins regain their biological activity by reversible process. That process is called renaturation. So in this situation, when temperature or the pH because of which the denatured has been brought about, when these conditions are changed, then the protein can be renatured. Renaturation of the protein. But the thing is, only some proteins can regain their biological activity. And when they gain their biological activity, this process is called renaturation of protein.